Howdy, my name is Jacob Tigarina, and the topic that I have chosen is Nosema ceranae and its effects on the honeybee lifespan. Apis mellifera and Nosema ceranae. Apis mellifera, otherwise known as the western honeybee, was imported to the United States by European colonists as early as the 1600s. These bees have become known as a major source of pollination within the United States and play a large part in the agricultural community. Nosema ceranae is a fungal parasite that has targeted the Asian honeybee, but has now been shown to start to target Apis mellifera as one of its hosts as well. This fungal parasite is known to drastically reduce the lifespan of bees, killing them prematurely. It also spreads through the digestive system of the infected bees, which can contaminate food sources the colony may depend on. This graphic on the right is from one of my sources, basically showing a time chart of the spread of these honeybees throughout the United States. On the right hand side, there's earlier dates, and as it moves progressively towards the left of the United States, the dates become later in time, just showing the eventual spread of these honeybees. Our question: how does Nosema ceranae affect the lifespan and longevity of honeybees? Development within the host. After a bee has fed off a contaminated food source, this bee will soon become infected with Nosema ceranae. The parasite will then travel towards the midgut of the bee, where it will attack the host's epithelial cells and begin development of spores within the host. As Nosema ceranae develops within the host, it will feed off of the nutrients that the host would normally gain by eating. By robbing the host of these nutrients, the host will have an elevated appetite and will eat more than their usual amount in an attempt to compensate for the loss of nutrients that is being consumed by Nosema ceranae. This figure on the right is from a control group and an infected group that has been infected with Nosema ceranae. The control group is the dotted line, the infected group is the solid line, and this shows sucrose that is consumed by both groups over a 24-hour lifespan. And you can see through the chart that the infected group is actually consuming more as a whole compared to the control group. Killing the host. As mentioned before, Nosema ceranae is known to rob the host of nutrients, but not only that, it is starving the host to death and stressing its body as well. In 2007, Higgs et al. Conducted a study that involved two groups of honeybees: a control group that was not infected, and the experimental group, which consisted of honeybees that were infected with Nosema ceranae. The study seemingly showed that no matter what their experimental conditions were, the bees that were infected with Nosema ceranae had all died before or on the 8th day of the study. By parasitizing these bees, Nosema ceranae is causing them to die prematurely and shortening their lifespan. The figure on the right is from the study that's mentioned in the second line of text. This is the infected and control groups, the percentages of mortality, and the percentages of the infected bees as well. And you can see during the 6th and 7th days, that's when mortality was the highest, and usually, again, during the 8th day, no infected bee was left. Killing off the colony. If left unchecked, Nosema ceranae may be able to kill off entire colonies by infecting individuals through bees feeding one another or by feeding from infected food sources. Nosema ceranae has also been shown to seemingly expedite the age polytheism process, with nurses transitioning to foragers faster than expected. With infected bees foraging and bringing back food, this can lead to widespread infection. A study conducted by Martin Hernandez et al. In 2007 found that there was a significant causative relationship between Nosema ceranae and the development of hive depopulation, showing that this parasite can lead to large-scale damage. This figure is from that same study from Martin Hernandez, showing a, a percentage of positive samples for Nosema spanning from 1999 all the way to 2005, showing that there is a trend of these spores spreading at a pretty elevated rate. Conclusion. Nosema cerenae is a fungal parasite that has been targeting Apis mellifera and is leading to a shorter lifespan for these bees. By robbing the host of their nutrients needed for their survival, this fungal parasite is making these bees overexert themselves. The lack of nutrients being gained by the bees leads to the host eating more. This parasite spreads through the digestive system of their hosts and can contaminate food sources that other bees will feed on and become infected as well. With these parasites attacking the digestive system, this leads to the bees starving to death and leading to lower productivity, shorter lifespans, and colony depopulation. Significance. With Nosema cerenae leading to premature deaths in the western honeybee and the depopulation of colonies, this parasite needs to be studied further to find solutions to combat these declining numbers. If this parasite spreads on a larger scale than it already is, it can possibly lead to the extinction of Apis mellifera and the loss of a major source of pollination for the world.